All right, thank you, Dimitri, for that kind introduction. It's not every day that I get to be on the same session as my PhD advisor, so I got to be on my best behavior. Yeah, so I'm excited to talk about our project, Chips in Space 2.0, using microgravity to model inflammaging in complex organ chip models of gut, brain, and heart. This is a collaborative project with a number of folks at our Regenerative Medicine Institute at Cedars-Sinai, and co-PI is also Dr. Clive Svensson, the director of our institute. Let's see how I can move this forward. Okay, I'll just click the button. Oh, okay, got it. Okay, so this has been alluded to in a number of talks already, but there's been a dramatic evolution in the model systems derived from induced pluripotent stem cells over the past 20 years. In fact, this year is the 20th anniversary of the initial derivation of human-induced pluripotent stem cells by Shinya Yamanaka and colleagues in Japan. And so initially, of course, the initial differentiation protocols to transform iPSCs into different cell types typically relied on 2D mono and multi-culture, just two-dimensional adherent cells in a dish, which are, it's a high throughput approach, but perhaps not the most physiologically relevant, but scalable, and the protocols are straightforward enough that even undergraduates and high school students in the laboratory can execute these protocols. But there's, like I said, a dramatic evolution in these protocols to make these iPS-derived model systems, and as was just alluded to in the previous talk, now there's three-dimensional approaches, organoids, assembloids that are better physiological representations of not only what happens terrestrially to organs of the human body, but also potentially in space as well. Now, our laboratory and our institute has done a lot of work using microfluidic chips and organ-on-chip systems over the past 10 years or so, and I'll highlight some of the studies here. A number of these studies have been conducted within the confines of the Emulate organ-on-chip system, which has been alluded to already during the symposium. It's a powerful system that allows for multiple vessels, multiple compartments that enable the simultaneous growth of different cell types. For example, you can grow, cardiac vasculature simultaneously with cardiac myocytes, mix and match, with different neural cell types as well. And just highlighting on the bottom left of the slide there, a number of the publications that we've published in our Regenerative Medicine Institute using this technology. For example, Clive Svensson has published on a brain-on-chip system looking at blood-brain barrier permeability with this Emulate organ chip system using brain microvascular endothelial cells combined with neurons on separate channels of this chip. My lab, and I'll talk about this in a little bit, recently published a heart-on-chip system looking at cardiomyocytes, heart muscle cells on one side of the chip, and endothelial cells, blood vasculature on the other side of the chip. So it's a powerful system in part because it's an active biomechanically system that's able to be stimulated biomechanically, which allows us to actually mature iPS-derived cells pretty effectively by active flow of media or biomechanical stretch on the system as well. So alluding to that multi-lineage heart-on-chip system that we developed recently, this is work led by Maida Mosneb in our lab, again, using the Emulate S1 system. She was able to grow on the two separate channels, cardiomyocytes or heart muscle cells, and on the other channel, vascular endothelial cells derived from iPSCs. And we were able to demonstrate that there was actually a true advantage to growing these cells in this format as opposed to two-dimensional adherent monoculture, with the big advantage being the improved maturity of the iPS-derived cells under biomechanical stimulation over the course of multiple weeks. So this is a representation of that. So you can see just in the unstimulated, non-biomechanically stimulated condition on the left, you have cells that are growing somewhat synchronously and they're contracting somewhat in a synchronous fashion. This is actually a GCAMP calcium reporter line that allows us to look at calcium flux in these functionally active cardiomyocytes. So you can see that green flash as an indication of the calcium activity. And you can see on the right, we have a version of the chip that's been under active biomechanical stimulation for 10 days. And you can see a really nice synchronized flash of calcium in that particular heart-on chip. So that is just a representation of the advantage of growing the cells in this format. And then we applied this heart-on-chip system to studying drug cardiotoxicity. So this is an example of a drug called sorafenib, which is a known cardiotoxic agent, tyrosine kinase inhibitor, being used for treatments for various types of cancer. And we're able to show multi-lineage toxicity on both endothelial cells and cardiomyocytes growing within the confines of this heart-on-chip system. So that's one application of this, this heart-on-chip system. So why space? And a lot of this is kind of rehashing things that folks have already talked about. As we know, humanity is going to be spending more time in space in the years to come. There's a dramatic growth in the commercial space industry. We're going back to the moon. We're going to go to Mars. And certainly we know what happens to the human body at the macro level. We know there's, as Joe alluded to and a number of folks alluded to, there's cardiovascular deconditioning, musculoskeletal atrophy, changes in the immune system, and hematological changes as well, neurocognitive dysfunction that was just alluded to, GI dysfunction. And these kind of combine to mimic what's considered an accelerated aging phenotype, right? So we know what's happening at the macro level somewhat. Now, how do we distill that to the micro level, to the cellular level, and how do we actually model those cell-based changes? So stem cell research, indeed, has been happening on the International Space Station for a decade plus now, in large part thanks to support from the International Space Station National Laboratory, CASIS, NASA. These are just some examples of NASA astronauts executing some of our projects on the International Space Station, as well as some other projects as well. And Joe alluded to this as well. About 10 years ago, when I was a grad student in Joe's lab, I had the opportunity to conduct this project, which is the first long-duration cell culture experiment conducted on the International Space Station, and this was in partnership with Bioserve and Stephanie representing Bioserve in the back. This is a relatively straightforward project where we're growing two-dimensional adherent cardiomyocytes for a period of 30 days aboard the International Space Station, and then characterizing them using contractility analysis, structural analysis, and genetic analysis as well. And perhaps our greatest partner in these studies was astronaut Kate Rubins, who is not just an astronaut, but also a biomedical scientist in her former career. She was a PI at the Whitehead Institute before she got tired of writing R01s and decided to become an astronaut instead. So she was a tremendous asset and help in this particular project. So fast forward 10 years later to 2026, Cedars-Sinai, our institution, has been very active in this space, pun intended. We've had about 10 different missions that have flown to the International Space Station, looking at all aspects of stem cell growth, stem cell culture, automated cell culture in space, in partnership with our on-campus biomanufacturing facility that makes thousands of different iPS lines for internal use and external use at Cedars-Sinai. We've recently produced iPS-derived organoids from scratch, and for the first time in space as well, basically harnessing microgravity to make three-dimensional tissues in space. We also have recently made induced pluripotent stem cells in space, so we are really excited about the prospect of harnessing microgravity to potentially make things in space that you may not be able to on the ground, and also to model human physiology as well. And so talking about our organ-on-chips study, a project that we're fortunate to have just gotten funded, I'll talk a little bit about that. So here we are modeling cellular aging in the brain, gut, and heart using these chip systems that I have alluded to. We know that cellular aging leads to functional decline and susceptibility to age-related diseases. We know that monocytes and immune populations play a significant role in age-associated inflammation, also known as inflammaging. And as has been alluded to, these space conditions can mimic inflammaging and actually enable the study of aged immune cells on these different microphysiological systems harboring different organ analogs. So we're going to develop different MPS models on the International Space Station to model the impact of spaceflight-induced tissue degradation, aging, and inflammation, with the focus being on the heart, the gut, and the brain. And I'll talk a little bit about some of the preliminary data that we've generated for the grant already. Certainly, we've had a lot of expertise, as I've alluded to, in developing these different chip systems. We've had a brain MPS harboring iPS-derived neurons and glial cells, a gut MPS with iPS-derived intestinal organoids, and all these are within the Emulate chip. And then my lab has developed a heart MPS with iPS-derived cardiovascular cells. We also have, and like I said, an important part of this grant is the immune component. And so Ali Moser and Clive Svensson's lab has developed a protocol for creating iPS-derived resident macrophages that we're able to integrate into each of these chips. These are the different milestones in the grant itself. I'll just highlight a few here. The first was to demonstrate the survival and functional stability of these different MPS models over multiple months in these in the emulate chips, over 6 months actually. So we've never actually grown the chips for that long, and we've had to be creative in how we get to that point. But I'm happy to say we are close to that point. We've been able to grow these hard-on-chip systems for close to 5 months now, as well as the brain-on-chip as well. The gut-on-chip system has been a little trickier given the high proliferation rate of those organoids, but we're confident that we'll be able to get to that point as well. And then we've introduced the macrophage, iPS-derived macrophages, also known as IMACs, into each of these chip systems, the gut, the brain, as well as the heart system, to see their integration and proper functionality, which we're in the process of characterizing. And then, shout out to Stephanie and the BioServe team who are developing systems to support MPS culture on the space station. This is work that's actively in progress, and this is highlighting some of the chip systems that they have developed in the past, and we're hoping to adapt the emulate chips to this hardware as well. And with that, I'll just wrap up and acknowledge the folks who have been critical to getting things off the ground, pun intended again, and especially my partner in crime, Dr. Clive Svensson, as well as folks from across the Regenerative Medicine Institute at Cedars-Sinai. It's really a very collaborative environment. So thank you so much.
Thank you, Arun. Are there any questions for Arun? Well, I guess I can ask a question. So you experienced different transports and different missions, and some of them are private, I think Axiom. What are your overall impression of those? How different they are? What are the benefits of one over the other?
Yeah, I mean, they've been pretty comparable. We've flown missions with Axiom Space, for example, like you alluded to. The execution of the projects has been pretty comparable in part because we've had great partnerships with BioServe. I think there's an advantage to having certain mission specialist astronauts, as Mike can understand as well. I have folks who have biomedical training who would be able to kind of speak the language of spaceflight as well as the language of biology. And I think those have been our most successful projects, the projects where we've been able to work with folks like Kate Rubens, Peggy Whitson, astronauts who have that biomedical background. Yeah.
Thank you, Arun.
Yeah.
May.
Yeah, I just have a quick question about the heart-guard brain. Are you going to have individual organelle model for each one, or that they're interconnected? So just like what's the rationale to have heart-guard brain as the triangle?
Yeah, they're going to be separate systems. The multi-lineage part of it is going to be the integration of the macrophages. So that's how we develop that multi-lineage context. But what we're also hoping to do as part of the grant is to see if we can develop an all-in-one media that would be able to potentially connect these different chip systems if we need to. So a hybrid media component. So yeah, that's part of what we're working on.
All right, thank you.
Thank you, Arun.
Thank you.